Caffeine as a biological switch. The future of precision gene editing. New research from Texas A&M University has introduced a groundbreaking concept in biotechnology, using caffeine as a chemical on-off switch to control gene editing tools within the body. While this is not an immediate medical treatment, it represents a significant leap forward in the quest for precision medicine. The ability to target specific cells without causing widespread systemic side effects. The innovation. Caffabodies, a research team led by Dr. Yubin Zhou, has developed synthetic proteins dubbed caffabodies. These proteins are engineered to remain dormant until they encounter caffeine. Once caffeine is detected, the caffabodies trigger the assembly of components required for CRISPR gene editing. The mechanism is highly sensitive. Activation. Only about 20 milligrams of caffeine, roughly 1/5 of a standard cup of coffee, is required to trigger the process. Deactivation. Once the caffeine is metabolized and cleared from the bloodstream, the gene editing process naturally ceases. To provide even greater control, researchers also developed a secondary, off switch using rapamycin. This dual control system allows for more granular management, enabling scientists to shut down the editing machinery faster than the body's natural metabolism would allow. Why caffeine? In the field of molecular biology, finding a trigger molecule is difficult. Most chemical triggers require specialized drugs that can be expensive or carry their own toxicities. Caffeine offers 3 distinct advantages. Proven safety profile. As one of the most widely consumed substances on Earth, its effects on human physiology are exceptionally well documented. High accessibility. It is easily obtained through common sources like coffee, tea, and chocolate. Natural clearance. The body has an efficient, built-in way to process and remove caffeine, providing a natural way to terminate a biological process. Potential medical applications. The most immediate impact of this research lies in immunotherapy, specifically CAR T-cell therapy. Currently, CAR T-cells are engineered to hunt and destroy cancer cells. However, a major clinical challenge is that these cells are always on. This can lead to dangerous complications, such as cytokine release syndrome, where the immune system overreacts and causes severe systemic inflammation. By using caffeine-controlled caffabodies, doctors could theoretically activate these cancer-fighting cells only when needed and deactivate them once the threat is managed, significantly reducing side effects. Beyond oncology, the researchers demonstrated that this system could potentially manage other chronic conditions. For instance, it could be used to trigger controlled insulin production in patients with diabetes, offering a level of precision currently unavailable in standard treatments. The path to clinical use. It is important to note that this research is currently in the proof-of-concept stage and has been conducted in laboratory settings, not in human subjects. Before this technology can reach a pharmacy, it must navigate several rigorous hurdles: extensive long-term safety studies, multi-phase human clinical trials, strict regulatory approval from agencies such as the FDA. Conclusion. This study marks a creative milestone in gene therapy, demonstrating that common, everyday molecules can be repurposed to control sophisticated genetic tools. While your morning coffee won't cure cancer today, this research paves the way for a future where medical treatments are more controllable, safer, and highly personalized.